Welcome, Wordsworth College and friends. I would like to start this evening with a statement of acknowledgement of traditional land. We wish to acknowledge this land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. On behalf of the Wordsworth College Alumni Association, I am very pleased to welcome you this evening to our first Alumni Cafe event of the year. Tonight, Professor Becky Sigmund will be presenting a talk entitled From Fossils to Genes. Professor Sigmund is a paleoanthropologist with research interests in early human evolution. She has researched the origins of erect bipedals and hominid fossils in the East and South Africa, as well as expeditions to Ethiopia. Professor Sigmund lived briefly with the San hunter-gatherers of the Kalahari Desert for the purpose of conceptualizing the nature of early human subsistence patterns. Long-term interests are what is human, as reflected to of Sigmund's books, Human Ecology and Challenges in the 21st Century, and Physics, Evolution, God, Mass and No Mass. Professor Sigmund, you're welcome to.
Okay, thank you very much. And thank you for inviting me to give this talk. I'm assuming that everything is okay and we've started. All right, so I'll begin my talk. I wanted to begin by talking about my working definition of fossil and gene, which will fit into the talk I'm giving today. So to begin with, a fossil is an unbiased record of our past, our evolutionary origins based on preserved remains usually found in the sediments of Earth. A gene, on the other hand, is a record of one's own personal heritable biology and the relationships that exist between that person or a group of people, amongst individuals, amongst families, and amongst populations. Secondly, there's some very interesting comparisons and contrasts between the study of genes and the study of fossils. And I'd like to take a brief look at what those are, because they're quite relevant to understanding how the disciplines differ, as well as how they are becoming a little bit more similar. The time that paleontologists deal with is, or may be, extremely long, and today's talk will be dealing with the time of around 4 million years to 1.5 million years, and I'm defining that as the earliest hominid stage of evolution. The time that geneticists work with are primarily a lifespan, and for individual people, that is about 100 years, more or less, at least possibly 100 years. Although the time that geneticists are able to deal with now is beginning to be changed because of their introduction of a new specialization, ancient DNA, which we will discuss toward the end of the talk. Historically, paleontology and genetics have proceeded in somewhat similar societal waves because it's society that has an influence on our belief systems. I will head back very quickly to 1850s, the middle of the 1800s. This was a time of Darwin when his big book came out on the origin of species. This was a time that the first Neanderthals were found, and this was a time that Mendel discovered what we are now calling Mendelian genetics, the passing on of hereditary units in plants to the next generation, and what's the basis of it. So that was all happening in the 1850s. very interesting correlations. Both were being very active, but Neanderthals, which were the first fossils to be found, were rejected. People said they're not our ancestors, they're abnormal, they're pathological. We don't believe that we had an evolution like that. So there was also a lack of interest by science. With Mendel's work, there was simply a lack of interest. People were not really concerned. And so it sat in his publications until about 2 generations later, when Mendel was rediscovered. And this was about the time that paleontology of humans got started again. So that's a similarity and an overlap there. Genetics was rediscovered around the turn of the 20th century, the early 1900s. And then it continued, especially by embryologists who took 2 different pathways of looking at the inheritance of traits and embryological development. And this is where the genes start doing their work. A visionary embryologist by the name of Waddington coined the term epigenetics, which now has become popular again, but for several generations it was rejected because it rang of Lamarckianism, that is, the environment influences the genes, and that was not what was considered by the other school of thought, where environment's just kind of noise. We are concerned with one gene, one trait. Well, consider we're the ones who make up what the traits are. How do the genes know that? Anyway, that's the way that genetics went for some period of time. Now we start to get together again in the 1950s. Mrs. Please, and that's in one of my descriptions of this talk. Mrs. Please was and is a fossil. I'm not sure if you can see, but I'm holding her here. And we will be looking at some slides of her later. This is a cast, obviously. The original is in South Africa. The importance of Mrs. Please is that it was the first time in the history of paleontology that it was recognized that we ourselves have an evolutionary past. And when I get to the slides, which will be shortly, I will describe a little bit more why she was so important and what traits that she held that had never been seen together before. What was happening in genetics at this time in the 1950s is another very big event, the identification of DNA, which is a term everybody knows now. It's become so popular in our gene-centric biological world now. So this was a similarity. There were 2 major events occurring both in genetics and in paleontology. Paleontologists always included the environment in their studies because it's the marker of evolution. And environmental studies were always a part of paleontology. But environment, as I pointed out before, was not a part. And it wasn't, again, interestingly, until the end of the 1900s that epigenetics, now called the new epigenetics, was discovered not by geneticists, but mostly by medical scientists in their work. Now, one last thing about the difference between fossils and genes is that there are rules in paleontology for how to go about naming new specimens. There's what we call the Code of Zoological Nomenclature, and that gives us rules by which we must or should follow. Sometimes people are not educated enough in that. And so we have a large number of names of fossils, some which will eventually have to be erased and put into other groups. There are no rules of how geneticists should do their work. And this may be leading to a problem. Those of you who have been reading about stem cells and some of the ethics and where the stem cells come from, And so that's the only rule that at the present time is present. So those are some of the interesting comparisons between genetics and fossil studies. One last thing I'd like to say, I have divided the evolution of our hominid family, and it's taxonomically a hominid family, based on one particular characteristic, which I'll mention when we get to the first slide. And I have divided our evolution into 3 stages. This would appall most paleontologists, but it's very appropriate for presentation of major traits in a field. The earliest stage is the one I'm talking about today, and it runs basically from a little over 4 million years in time to about 1.5 million years in time. All the fossils within this time range, and given the particular traits we'll be talking about today, are called the Australopithecines. Australo means south, and Pithecus means monkey-like or ape-like. So the very first one that was found was thought to be a monkey-like southern ape. And that's the origin of the name, and that's the origin of the first stage of human evolution. Just briefly, the second stage is basically Homo erectus, and it goes from 1.5 million to 200,000 years ago, and there are features that characterize it, not for this particular lecture. And the third stage is ourselves, Homo sapiens, which started at about 200,000 years ago, based on anatomically modern human skeletons. Okay, now I can move on to the slides. I think I can move on to the slides. Okay, got some problem here. Option control. Can somebody help me out here? I'm not able to change the slide.
Vinny, can you jump in here?
Should I put option enter? Is that what I'm missing here?
Vinny, can you advise?
Yeah, hi, sorry, I'm here.
Yeah, I'm just wondering. I'm not able to move the slide.
So can you, so option enter is not working, and the arrow keys are not working.
Okay, yeah, thank you.
There we go.
Okay, yeah, that's working. Sorry. All right. So here is the actual discovery of Mrs. Please. And again, I'm not sure how much you can see here, but compared to the shape of my head and the size of my head, a small woman, this is how big she is. It's a replica. When it was first discovered, it was blasted with dynamite out of limestone deposits. And the limestone deposits, fortunately enough, severed one piece that kept the entire skull intact, except that it divided it in half right at this particular point here. So what you can see, and if I can use my arrow, good. This would be the cranial cap, which is entombed in limestone, sort of concretion, and the bottom of the skull is right here. So the two had just been separated, and this would have been taken by the Johannesburg Star and published in their newspaper actually in 1947. Because before they did any further excavation, they called to record this fossil that is going to change scientific beliefs to make science realize that we did indeed have an evolutionary past, and we went into different stages of morphological development. The next slide shows Dr. Broom, who is pointing to the bottom of the skull. And in the 4 slides, you see that he is eventually, it's being taken out of the limestone concretions. And this is partly by physical force and partly by the use of acetic acids of differing concentrations in order to expose it. So this is a very significant discovery. It's what really changed the evolutionary thinking in human paleontology. It's it's kind of a magical one to me, and I have had the fortune to see it in real life in South Africa during my research periods. We can see a comparison here of a modern human on your right-hand side with Mrs. Please. Her actual notation is STS5 for Sterkfontein number 5. And the Mrs. Please, the Please stands for a name which is no longer in use, and that is Plesianthropus transvalensis. We don't want to call her that. Over the years, it's found that there is one primary genus, and that's Australopithecus, and she would have been the one that actually proved that there is this genus. And so you can see that the cranial height of the two indicates the size of the skulls and the size of the brain inside of them would have been very different. In actual figures, Mrs. Please has about 350 cubic centimeters of brain space, whereas modern humans on the average have 1,350. So she's less than a third the size. And this holds true for subsequent fossils that were found of her own species. A lateral view shows a significant discovery that not only was she small-brained, but that the brain came after the expansion of the brain came after the origin of erect bipedalism. The support that you see at the back of her head is indicating that the opening from the skull to the spinal cord is vertically directed downward. And again, if I can use the arrow, I'm talking about this area, this location here. And that is the same feature that we find in all human beings. We are upright bipeds. It was the first time that was discovered that erect bipedal posture came before the expansion of the brain. And this was, again, a monumental discovery. So this is the first of the major sites and fossils that I'm going to be showing you. You'll note when we look later on at sedimentary deposits that they are very different than the limestone deposits, which these fossils are found in. And it makes quite a difference also in the dating techniques. The next slide will take us to what I just mentioned to you. There is an opening in the base of the skull, and the vertebral column in human beings is basically vertical, whereas it's projecting back and it's concave or convex, depending on which way you look at it, in the apes that are the closest of our primate relatives. These areas, the skull and the pelvis, pelvic girdle, are extremely important in indicating what kind of posture an animal has. How do we know when we find a fossil that is going to belong in our human evolutionary lineage? Because we usually find fragments of it. Again, this is a complete skeleton of the gorilla on your left and a human on the right-hand side. And if we find the area that was found in Mrs. Please. It's the big hole, or the foramen ovale, which is a connection between the spinal column and the brain. And this comes out at an angle backwards in the gorilla and in almost all other animals, whereas it's vertical in human beings. The pelvis is extremely different in human beings than any other animals, and many of the other bones. Comparative anatomy is extremely important for paleontologists to know, because otherwise they are going to have problems in identifying animals. So comparative anatomy is what I'm emphasizing here. We spend years trying to learn the comparative anatomy of as many animals which are either closely related or not related, but might be in the sediments with the other individuals. And if you just find a fraction or a piece of a bone, it's sometimes very difficult to determine whether it belongs to a human being or a primate or a carnivore. And so we spend years learning this, and that's one of the ways that we can identify. We can also call in specialists who know, in this case, this would be horse skeleton, and so they would be specialists in horses and could tell us whether it's a horse bone or a human. But that's to come later. So to emphasize and to sort of summarize the importance of South Africa and its contributions in Mrs. Please, we see this reconstruction of an erect biped with an Australopithecus face. And there's a stone in his hand. Tools were not being used this early, not 4, not 3, but perhaps closer to 2 million years. Stones might have been used. The actual Mrs. Please is dated now at 2.5 million years by several different dates. And I will be pointing out some of the dating techniques as we look at the different situations. I'll be moving now down to the location of a very famous site in Ethiopia. It's the site where Lucy, a 40% complete skeleton, which was fossilized in just north of Addis Ababa. And it's the Rift Valley, which we see in the next slide, which is defined by these triangles here. The Rift Valley is a great area for erosion. The site is located in the Afar Depression. So during the times that the fossils were running around as living individuals, there would have been a great deal of geological and volcanic activity, so that there was volcanic ash that was deposited, sedimentary sands and so on were deposited in order to create the site where we're going to be going to just shortly. Paleontologists need the help of geologists, and it's the geologists that give us the information about the dating and following the stratigraphic layers out before we start picking fossils out of places when we are looking for them. They have to have a location before they can be dated. If they are on the ground and they haven't been dated, then we have to use some other means. But when they're in the sediments, we can date the sediments by radioactive dating. Flying over these sedimentary remains in Ethiopia, you see the serpentine line going down the middle, which is actually an old stream bed. And those are the places that one starts to look because of gravity. When fossils are buried and then they begin to be eroded out, obviously gravity pulls them downward. And so paleontologists tend to walk in areas like this looking for fossils at the bottom and then tracing their origins upward in these hills Most of the ones you see range to be about 300 feet in height. The next slide shows them at a horizontal level, and you get a very good idea of what it's like to be in the sedimentary deposits in the Rift Valley. In the background, the very last hills that you see here, you can see a white line going across here, and then another one above here, some down below. Those white lines represent volcanic tuffs, TUFF. A volcanic tuff is the ash that is left from the volcanic eruptions. And fortunately, everything chemically begins again, so we can give potassium argon dates of these volcanic deposits. I started to say carbon-14. Carbon-14 cannot be used in paleontology because it takes more than 60,000 years to fossilize, and carbon-14 can only date up to about 60,000 years. So the deposits I just pointed out to you, the white lines in the background, are going to be around the age of 3 million years. And the Lucy material would have been found just in or above or below some of these deposits at 2.8 million years. Paleontologists do a lot of walking before they start to find any fossils, and they do have to be well versed in some comparative skeletal biology. The site here that is being excavated and sieved represents one find that was made the year after Lucy was discovered. It's called the First Family, and it's called that because the fossils that were eroding from this area, actually they were found first down in the river valleys, and then we walked up the hill until we found the basic sedimentary deposits they were coming from, and started sieving to find smaller pieces of bone. So the sediments here are from the First Family. What is the First Family? It's a small population, possibly a family or a small group of early hominids that got caught in either a flash flood or volcanic, covered by volcanic ash by a volcanic eruption, because somewhere around a dozen individuals or parts of individuals were found, somewhere in the order of, I think, about 3 or 4 dozen specimens. And specimens can be just a fragment of a femur or a tooth when they are found separately and not in the bone itself. But it was enough to say that there were around 10 or 12 individuals, including some children, and they were all buried at the same time, 3.1 million years ago, and have been entombed in the sediments until the expedition came and found them. And this makes one aware, if it takes so long for a fossil to form, we really should treasure them as something that we cannot make ourselves. Our lifespan is too short. We cannot make it. It's something that nature makes, and it's something to marvel at and to be very grateful for, to understand something about our past, which tells us something about ourselves. Now, if you were in Hadar and you were one of the paleontologists walking around and looking for a fossil, I've found this slide very instructive. Most of what you see are rocks, and they are either brown or very gray, but somewhere in that slide, there is a fossil. And you can tell because it just seems to have a slightly different look and shape. But the next slide is a closer view of it, and I apologize for the darkness of it. What you see are molars that are erupting from a hippopotamus skull. which is turned upside down because it's rolled out of the sedimentary layer that it's found in. It's eroded out. So we just go back and usually in an audience, I can see people, but I can't see you people. And I can get some reaction. I'm sorry. I have to pretend that I know you're there, but it's just kind of, Zoom is still kind of strange. Anyway, in the center of the picture, which I'm sure most of you have picked out already, is where this hippopotamus skull was found. Okay. We go on to look at this strange looking slide here. As I said previously, paleontologists are very interested in the environment. If you know something about an environment, you can tell how an animal is having to adapt to its environment. And the way it has to adapt has something to do with its morphology. It has to find food in the environment that it's living in. So environment and form and function go very much close together, which is something that it took geneticists to learn a little bit longer. But then they're very, very different disciplines. And the fact that they are learning now is quite amazing. Anyway, to get back to this slide, these are well-preserved crocodile eggs. And that's highly unusual. So that suggests that the water in this area was moving very slowly. Either it was a very still lake or a very slow moving stream bed for crocodiles to lay their eggs and for them to be covered with sediment and to fossilize over 3 million years. This particular site is famous for that because it's so rich in a variety of other fauna than just hominids. Well, you guessed it, this is Lucy. And we have enough of the skull, the mandible, and some parts of the cranium to see something about her diet because teeth reflect the diet of an animal. And we have the pelvis and the femur, which is definitely that of an erect biped. So once again, around 3 million years ago, or more precisely 2.8 million years as dated by potassium argon, this person lived. It's thought to be a female because male and female pelvises look different today, and they probably looked a little different 3 million years ago. Although that's a problem one has to consider as well. Are we comparing different skeletons with different morphologies if we use Homo sapiens? But that's a problem we were also starting to sort out. So this is Lucy, and she's famous because we can now correlate cranial features with upright bipedalism. With Mrs. Please, there was no postcranial material, so we had no pelvis, no femur. We only assumed that she was an upright biped based on the cranial features and the location of the connection to the brain and the spinal cord. After a day in the field, we're still at Hadar, and we've come back in the evening and or in the afternoon, and put our finds out on a table, and then try to figure out what they are. They all have been classified before removing from the sediment. We carry little notebooks around, and so you have to know what geological sedimentary layer they're coming from. And so these represent a variety of the animals that were present. There's only one fossil in there that is a hominid fossil. That's not the one in the center. The one in the center is a monkey. It's too prognathic and the skull is too small. But just right of it, that one with the big head of a femur is enough to tell us that it comes from an upright biped. So once again, know your comparative anatomy. Okay, that's my second major site that I think has been extremely important in guiding other people to compare their more recent finds with the first ones that were found that showed a combination of traits or a change in the belief systems. The entire Leakey family has been extremely prominent in human paleontology, and I have focused on Richard Leakey and when he was younger and very active in the field, and one of his good friends, a Kikuyu, at least you see that from the neck down, and what an Australopithecus might have looked like. I think it's a bit out of balance, but it still shows that if a Kikuyu had the body of an Australopithecus, it would be much better. But anyway, it's to show that Richard Leakey has been very prominent in the field and has encouraged interdisciplinary research. He has brought in not only paleontologists, but people who are specialists in different faunal remains, people who are specialists in environment, geologists he's brought in to his researches, and a new field that started developing around this time, taphonomy, the study of how the dead are distributed, are they found in place, or have they been moved by stream beds and so on, because it makes a difference when you're reconstructing what the environment is. So I just want to give credit to this person who's done a great deal of work in Kenya. And one of the deposits of his that was found is referred to as the KBS tuff, named after a taphonomist, K. Berensmeyer. This particular tuff gave problems to paleontologists because it did give two different dates dated from different laboratories. Now, science has to be reproducible to be believed. And these dates should have been the same because they were taken from the same sample that these geologists are taking up right now. The problem was there was contamination in one of the samples. Well, which one was it? One laboratory was dating the samples at 1.8 million years, and the other one was dating it at 2.6. That doesn't work in science. So more samples were taken, and other laboratories were included, and the actual date turned out to be the older date, 2.8. And so this is how science works, and that's just another example that I felt was important to throw in. We are careful, and there are a number of ways to date our material, not just one. And if it isn't right, then we know it's not right, and we try to make it right. A view of Olduvai Gorge, because now we're going to be heading down into Tanzania. This is what the environment looks like. And in front of you is 2 million years of deposits. Right in the center, you see the river valley, and then projecting up from it. This is an extraordinary paleontological location. And it has served as this sort of model and the base for comparisons of material from other locations. I didn't want to emphasize this because it's so prominent in so many things, but it has been extremely useful in paleontology. And give credit to Lewis and Mary Leakey, who discovered it and did a lot of work there. Now we're going to be moving further south from Olduvai Gorge, about 40 miles south of there. The Maasai still live in the Olduvai Gorge region, and the Maasai move around, a group of local indigenous people. They move around quite readily by walking 40 miles, no problem in a day. So one Maasai said to Mary Leakey, "We think we found something you might be interested in, just down here about 40 miles that we walked yesterday. Maybe you would like to know and would like to come look at it." And so Mary Leakey did make the trip to a place called Laetoli in Tanzania. And she's there being photographed when it was found. And what was found were hominid footprints, more hominid footprints and older than any that were known at the time. And these footprints have become so famous. Well, this is how the talk was advertised. So you've seen a picture of them, but they're absolutely phenomenal. How did they happen? Well, they cover a large area, and there were more than hominid footprints there. There were footprints of a number of different fauna. And they seem to be like so. These were the hominid footprints, 2 sets of them. And this is an artist's reconstruction of the hominids that are rushing away from the Sadaman volcano, which is erupting. And this gives you an idea of the situation that is 3.7 million years in age. So 3.7 million years ago, there was an eruption from a local volcano. These animals got caught in it: giraffes, various bird species, elephants. All of these footprints are found in this particular area. It's a fantastic location, and there's absolutely nothing else like it in human paleontology. The artist's reconstruction is simply that these footprints were made by 2 early hominids. And again, you get a view of what people, artists have or think that the Australopithecines look like. And I think the next slide, whoops, I missed that. This is the slide which shows that they were walking together in family style, and these were preserved and dated at 3.75 million years. So they're not necessarily the oldest ones now, but to find them in a situation like this is absolutely a marvelous discovery. Well, in summary of the 3 sites that I was talking with you about, there are the limestone caverns in South Africa, and the change in belief systems caused by Mrs. Please. And then there is the fact that nobody knew until Mrs. Please that erect bipedalism preceded the beginning of expansion of the brain, which continued into the later Australopithecus stage and into the Homo erectus stage, and then finally into the Homo sapiens stage. And it was the most noticeable continuous trend of increase in brain capacity that is present in this lineage. So that was Hadar, and then we went to east to Kenya, and then this. So these are some of the major sites that have made marvelous contributions to the field of paleontology and to human evolution. And in each one of them, we've wanted to know what the environment was. How did the environment influence the development of this particular lineage? And another very interesting phenomenon, which I'll talk about here, is the fact that ever since Mrs. Please was found, there has been sort of like a fossil gold rush. Paleontologists have wanted to go and find their own hominids. And so there has been a plethora of names. This probably represents that there was a great deal of biological variation in our lineage. And one of the things I'd like to emphasize is the fact that there still is a lot of biological variation in our lineage. They used to be called races. Now we don't use that term but the fact is there's a great deal of biological variation, and that's directly related to the environment that they are living in and trying to survive in. So the biological variation that's present in the early hominids can be representative of the lineage itself. And the next slide shows just a quick view of what these are going to become. It's either late Australopithecus or early Homo erectus. And these, being Homo erectus, are thought by paleontologists to represent our ancestors, the ancestors of all later Homo sapiens. The brain size is larger than those in the Australopithecus stage, and there are some other features as well. They're much more robust. They were beginning to move into colder climates and so forth. So that's just a very quick view of an artist's reconstruction of what the Australopithecus stage is going to be leading to. And then finally, I'm going to be talking just a little bit about Neanderthal. These are reconstructions of late Homo erectus or early Homo sapiens based on some Neanderthal remains. And I give you 9 different interpretations by 9 different artists. So this represents not only you've got a skull, but how you interpret what it looks like can vary tremendously. But the fact is there was really a great deal of variation, biological variation in our species. I think now we're going to be moving to ancient DNA. And I'll finish the talk off with a discussion of ancient DNA, what it is, what it may tell us, and what are some of the problems. Why do I use this? It's just to remind us that what DNA does tell us is relationships, biological relationships amongst families. I think most Canadians will probably recognize who this family is because of the English origins, Queen Victoria's family. And she's sitting there right in the middle at the bottom, Queen Victoria. And up to her left is a Russian Alexandra, who is going to be married to Nicholas. These were actually people who were done away with. But the important thing I want to show is there is a relationship, a genetic relationship, from the idea that there is one gene to one trait. And the trait that these women carried was hemophilia. Now, hemophilia, and we look at the family tree that's being drawn, has been made famous by this little boy here, Alexis, and the monk that treated him. He had hemophilia. Where did he get hemophilia? It has been drawn in this chart that it came from his great grandmother and possibly even one generation earlier because it's carried on the X chromosome. And I won't go into the detail of that, but it's what can be done with genetic studies. One can trace genetic relationships among families, individuals, and populations. That's just to remind you of the differences of genetics and fossils and fossil studies. So what ancient DNA is, and what you're looking at here actually, and this will be my last slide. I made it blurred because it's an old picture of Neanderthal. Not the first one that was found, but some later ones that were found. And the old-fashioned idea is that, well, these don't look like us, but, and besides, they look kind of slow and dense and so on. So poor old Neanderthal got a bad start. First of all, it was ignored, and then considered abnormal, and then considered really a backward sort of thing, which was a side branch maybe of human evolution. Well, what does ancient DNA have to do with this? Because somebody tried to find DNA in Neanderthal. Well, let's just backtrack a little bit. What is ancient DNA? Ancient DNA are bits and pieces of DNA in old bones and teeth that are found after death. Neanderthal was not the first that was studied for his DNA. The first was a 150-year-old indigenous American person, and it was done by a curious geneticist who said, I wonder if we can find any DNA in dead people, or how long it takes for the DNA to deteriorate or degrade or simply just fade away from existence. And so he did, and he found some bits and pieces of DNA in this 150-year-old indigenous American person. Then around the turn of the century, more curious researchers in genetics decided that they would start seeing if they could find DNA in other animals and in other individuals. And so the bits and pieces, why is it only bits and pieces, and why is there any at all? Well, first of all, there's not going to be any in Australopithecus, because Australopithecus is a true fossil. It is totally inorganic, and a true fossil is one in which the organic has changed through time into being inorganic material. So if there's no organic material there, then there cannot be any DNA. However, geneticists are still wanting to study fossils, and maybe the younger ones. Well, so this is what aDNA, or ancient DNA, is. Some of the problems with ancient DNA, in spite of the fact that it has been found in a variety of animals, just bits and pieces, it's very expensive to run the tests, and there are problems all the way along the line. First of all, it's destructive to the fossils or the subfossils and the bones. And because bits of bone has to be drilled out of the teeth and the bone of the fossil or the subfossil. Another problem, and a really big problem, is contamination. The samples that are in the soil are already possibly contaminated by fungi or bacteria. And at any step along the way, there may be contamination as the sample is being moved from the earth where it was buried into the laboratory, into the chemical tests that have to be done on it, and so on. And one very interesting example of this is some person thought he had some dinosaur, now that's very old, a dinosaur DNA. And when he tested and looked at it, he was very curious to see what it was. And what he saw is Y chromosome. In other words, the laboratory worker himself was testing his own DNA. So the contamination is extremely touch and go with these bits and pieces. There has been a sample of a Neanderthal in which apparently the entire genome was done, one at about the age of 38,000 years. And I have not read too much about that. It's a very rare find, and to date, something that old is extremely touchy. But it has been published, and so the comparisons now have been made between Neanderthal DNA and human DNA, and you can look on the internet and read about this and how you can determine how much DNA you have, which is related to Neanderthals. I think ancient DNA studies have perhaps a long way to go because of some of these problems of which I mentioned. One new problem that has occurred very recently and which I read about is that with the longer something is lying dead and in the earth, the more degraded becomes the DNA. Most of the time, it's almost impossible to find. So the sampling just goes without any results. But it has been suggested that the DNA that is in the bodies of these older specimens, the DNA itself, if there is any left, may be mutating. So if that's the case, then comparisons between Neanderthals or any older specimens with modern specimens or modern species is perhaps touch and go. Well, I've tried to highlight for you in a very quick fashion something about how fossils and genes do have something in common, and with the development of new methods perhaps for aDNA or finding more subfossils, we may be able to make relationships, as the DNA researchers are, between populations and possibly even diseases. But one of the problems with diseases is there's a great variety in diseases. Even hemophilia, there are several types of hemophilia. And so this may prove to be some problem as well. I'll conclude with saying and emphasizing that there always has been a large amount of biological and morphological variation in human beings and in our evolutionary lineage. And that is a very good feature to have because it's the source of the continuation and the continuing evolution of our species. And my last conclusion is that one of the best things I think that we can do is to work together and learn a little bit more of each other's disciplines, geneticists learning a little bit more about anthropology and vice versa. And with improving technology, perhaps ancient DNA studies will also become a little bit more developed. And they're just kind of struggling now, but that's the way with any good science. So if we can work together, we could contribute a great deal of information to what our species is all about. And this may be too powerful tools. These may be too powerful tools for the further evolution of our species. Thank you very much.
Thank you, Professor Sigmund. I will jump right into the Q&A session. And just a reminder for everyone, if you have any questions, just put them down below in the Q&A icon, bottom right of your toolbar. So the first question is, I believe it's about Mrs. Please. The question asks, have they found or theorized the reason it was only the skull and not the entire skeleton that was found?
Yes, there is a possible answer to that. Teeth are, there are certain bones that are thicker, more mass, and so they tend to preserve better. Some bones, like the ribs, are very fragile and so on. And so that's one difference in the morphology of the skeleton. You tend to find more teeth and you tend to find more of the hard bones. And that's why the head of the femur, for example, why would you find the cranium? Well, the cranium is not that thick a bone. That's just happens to be good luck in this particular case. And so aside from that, it's sorry, there's one other feature, and that is there has been more cranial material found before we started finding post-cranial material, because that's what people are looking for. And when they start finding what they're looking for, they will identify that first. There may be a femur lying beside the skull, but they're going to go directly to the skull instead. And so part of it is a behavioral response from humans liking the skull. We talk about that more than we do talk about the foot or the pelvis. As a matter of fact, when Richard Leakey decided that they needed to know more about the postcranial material and to find out what the morphology was. And they showed the people who are working, this is what a bone looks like of the lower limb, this is what a pelvis looks like. And so they started studying that. And instead of looking for heads, they started finding pelvises and femora and so on. So it's a noticeable, what are you looking for? And that's what you're going to see.
Thank you. Next question is it possible that more of Lucy's skeleton is out there somewhere? Or is it more likely that for one reason or another, they were not preserved following Lucy's death?
I don't think there would be any more of Lucy's skeleton, because there was a lot of excavating that was done to see if they could find more of her. So it was just some parts that were eroding out, and these were the ones that were found. The material was sieved, like you saw in the slide. And if there had been any more material, I think they would have found it. So we didn't find any more of her. But of the first family, again, there was a great deal of work that was going on to see if any more could be found in and around that area, and that it could not be. And if you find something that's further away, you can't identify it as being, a part of that particular family. You could identify the time, but not of that family or that group of people.
Thank you. Next question. Do we have evidence of extinct diseases or of earlier iterations of diseases found in the specimens of early hominids?
Not of early hominids. We do not. No. That's a simple answer. In the latest one, in later remains, we can, and this is true in possibly in some of the Neanderthal. There may be some diseases that they had that we have, but not in, there's nothing comparable that has been found in the earliest stage of Australopithecus evolution.
Next question is, I was wondering what your quick opinion is on degeneration theory rising in the 19th century, and if there are physical or physiological evidence.
The degeneration theory, I'm not sure.
Okay, so whoever submitted that question, maybe they can elaborate a little bit.
What you mean?
Okay. There's a question, it sounds like perhaps some career advice. Someone was asking, in your experience, what do you think is the best way for an individual to get involved in the ancient DNA field?
I think you would have to go to graduate school in genetics, and you would have to learn your basic genetics and then opt for studying with somebody, and there aren't that many people yet who are doing ancient DNA, but it would take a lot of background in genetics itself to do that, and some good luck to get in with the right crowd.
Another question is, have there been any similar finds as those in Africa in other countries, specifically matching the periods you spoke about?
That's a good question. There have been some questionable finds which had been made in East Africa of one of the Australopithecine groups, but it's very controversial. So primarily the very earliest stages, they are only being found in South Africa and East Africa at the present time. When we're getting up to the later stages of time, however, 1.5 million years, we're already beginning to get into some of the transitional forms. And so by that time, they are beginning to be in the old world, in Asia especially. Again, it's not my area to know specifically, but there are some that are found in the old world as well. They got out of Africa.
We have another question about disease. So the question asks, do forms of diseases correlate with environment or with biological evolution? In other words, does disease correlate more with genetic makeup of specific groups of races living in specific environments?
We probably have that answer in the past year and a half. There are some people who seem to be more immune to our pandemic problem, and some who are less because of the variation in individuals. But I don't know if that answers the question, not just by a comparison with the results of what's happened with COVID. Does it?
They will have to let us know.
Well,
another question is, it sounds like you are very doubtful of ancient DNA. Have the methods not improved?
Yeah, I didn't, I was trying not to be opinionated. It's just I've been reading so much about the problems, even by the geneticists themselves. And some of the scientists who are saying things like they're feeling threatened about their fossils. They're saying that there should be rules and regulations to protect our fossils from the research by the people in doing ancient DNA. Otherwise, they'll begin to look like Swiss cheese. And that's because you have to drill out samples of them from the teeth and from the fossilized bone. So if you're going to be doing that, it's a destructive technique. And the second aspect of that, which was pointed out in an article I read recently, is that the study is non-reproducible. You can't do it twice on the same Neanderthal. And you can't reproduce the results, which is one of the mandates of scientific research. So I hope that answers the question.
Thank you. We had a follow-up message from the individual who asked about diseases. And he says, I think this answer will have implications for DNA research, whether it is worth it on specific cases or not, I think. But thank you so much for the first answer. So some follow-up thoughts. So I think we will conclude the Q&A session now. Thank you very much, everyone, for submitting your questions. I'm sure Professor Sigmund's email inbox is open to future correspondence. So, Erin?
Okay.
I will.
Great. Hi, everyone. Thank you, Sophia. So Dr. Sigmund, on behalf of the Wordsworth College Alumni Association and on behalf of those from the alumni community who were able to join us tonight, I would like to thank you for taking the time to speak with us this evening. I'm sure I'm speaking on behalf of everyone when I say that I will be leaving this Zoom lecture knowing more about fossils and genes, and I'll definitely be having more questions and an interest in this topic. I'm sure I'll be Googling things later. As you know, as many of you know, the Alumni Cafe series is the Wordsworth College Alumni Association's flagship lecture series, and we have been holding these for years. I'm very happy that we have been able to continue holding these lectures during the pandemic, and the Alumni Association is very thankful to experts like yourself, Dr. Sigmund, for participating and allowing us to continue these learning events. So one last time, I thank you, Dr. Sigmund, for sharing not only your expertise, but also your time and patience to educate the alumni community and answer our questions. Thank you.
Thank you very much for inviting me to participate.
Absolutely.
Okay, I guess we all leave now.
Yeah, thank you, Doctor. Thank you, Professor Sigmund.
Okay, you're very welcome.
And thanks to everyone for joining us.
Thank you, everyone. Thanks for coming. Thanks, Sophia.
Thank you.